Welcome everybody to Betong Bungalow. It's really great to be here this evening, and this Betong Bungalow live show is being held on traditional land, the land of the Ngunnawal people. I'd like to acknowledge and pay my respects to elders past, present and emerging, and I'd like to thank them for caring and sharing the land we learn and live on. And tonight, joining me in Betong Bungalow is a very special professor. But before we get into that, let's just talk about poo, because if you spend any time around wildlife ecologists, or come to think of that, any kids aged 3 and up, you will quickly discover the fascination these two groups have with all things poo. And that is the absolute genius of this citizen science project we're talking about today on Betong Bungalow, a project that brings together kids and families and scientists to tackle antibiotic resistance in the wild. It's called Scooper Poop. The concept is simple and one my guest is going to tell us about today. Associate Professor Michelle Power is from the Department of Biological Sciences, Macquarie University. Michelle looks at host-parasite interactions in wildlife and how human disease gets spread to animals. Now, Michelle, you have a very impressive CV which talks about your special interest in protozoan parasites, endemic microbiota, reverse zoonoses, but we all know your favourite title has to be leader of the Scooper Poop team.
Yes, indeed. I think that at the moment it's the Scooper Poop project is one of the dominating parts of my work days. And it's super exciting because it actually gets me out and about to schools and community groups and engaging with the community about a global issue, which is fantastic. Everybody here at the Woodlands and Wetlands Trust headquarters has been really excited, and people coming to get their kits are showing lots of enthusiasm. Before we get into the nitty-gritty of the Scooper Poop project, how come poo is so crucial in giving us information about native wildlife? Yeah, poo's a great thing. It's actually a easy to obtain sample without actually trapping the animal. But in poo, stages of parasites are found, and also remnants of the bacteria that are in the gut. So we can actually look at the microorganisms living inside our wildlife just through a scat sample or a poop sample. Yes, I know that people, when you can't find the animals, you can use them even to count populations. So yeah, you can. Yeah, so the shape, size and colour of scat samples is indicators, or you can use those criteria to actually determine who did the poo. And how did it come about that we found out that human disease could be passed back to animals? Yeah, it's interesting. A lot of effort tends to go into zoonoses, so diseases coming from wildlife or other animals into humans. But it's kind of a unique area, is thinking about the wildlife perspective, and, you know, pathogens know no boundaries, or disease agents or parasites know no boundaries. And if they have a broad enough host range or can infect different animals, then they can move between both. And so we're interested in, the reverse. So our organisms going from us into wildlife. And why is that a problem if they start to get that antibiotic resistance? Yeah, so that's one of the things we're trying to understand. So might go back and just explain what antibiotic resistance is. Yes, first. Please, that would be great. Yeah, so bacteria over the years have evolved the ability to resist antibiotics. And they've done this because, you know, in a microbial community, bacteria need defences against other bacteria. And so they naturally produce some antibiotics. And then humans came along and discovered the power of these compounds to treat human diseases. And then, you know, things compounded, we flooded the environment, and we select for those bacteria that were defending against other bacteria through our use of antibiotics. Now, antibiotics are very important. They are particularly important for human and wildlife health. They're used in, wildlife and also our pets. So, but now what we've done is we've created such an really environmental pollution and spread those resistant bacteria into the environment and then onto wildlife. How it's impacting them is what we're trying to understand. And so how do they pick it up? Because the animals, how would they pick up that, what sort of pollution are they picking it up in? Yeah, so that's also part of the big questions, because it's really only in the last, 5 or 10 years that we've started looking and coming from what we would call a One Health approach, where to understand the transmission of any disease agent, we need to think about humans, wildlife, domestic animals, and the environment around us. So the general consensus across the scientific community around the world is that it's water pollution. So we're passing microbes in our feces down the toilet, sewage, and then the resistant bacteria and there are also the genetic mechanisms that enable them to resist antibiotics into the environment. And then our wildlife come along and drink water and may acquire. That's what we think's happening, but we're still collecting data, and so are other people as well, to understand that.
Yeah, right. So it's a really new area of science. So do we know what animals are being affected most at this stage?
Yeah, so in Australia, from our work, we tend to see quite high levels. So we've looked at a number, I'd say we looked at 10 or 12 species. So the Tasmanian devils, and that's from the land to the sea. So Tasmanian devils, possums is the big citizen science study. We've looked at some kangaroos and wallabies, echidnas, little penguins, Australian sea lions, and we tend to find them, and also bats, flying foxes, that the highest frequency of resistance genes, or our detection rate, is in the possums and also echidnas, which is interesting.
So, and the possums are going to be crucial in our Scooper Poop project that you're working on. But before we get to that, is there anything we know at this point that we can do to help stop human disease getting to the wildlife? We're talking about, our sewage, but things as citizens at home, as general public. Do we know anything we can do at this stage?
Yeah, well, generally, it's all about, a term we would use called antimicrobial stewardship. And then the second is about preventing disease transmission. So antimicrobial stewardship, that more sits with doctors and vets and people who prescribe antibiotics, and that they should be prescribing them at the right time.
So not just going in and saying, I've got a sore throat, give me those antibiotics, off we go?
Yeah, because antibiotics only work for bacteria. So if you've got a virus, they're not going to do anything. And I think for a long time we've had the consensus that if you are a bit sick, we'll give you antibiotics because it will help. You leave the doctor with antibiotics and you feel like you're going to be okay.
You're doing something.
Yeah, but so at that kind of prescription level, it's important there. It's also important for us as, I'm a stakeholder in treatments for disease. It's also important for us to understand that antibiotics only work against bacterial infection. And so we shouldn't be expecting antibiotics unless the doctor has diagnosed a bacterial infection. And then at home, if we do need antibiotics, and obviously we all will at some stage probably, but we need to take them until the course is finished. Don't stop when we feel better. And also make sure that we don't keep them and take them later or share them. And then equally, the last thing we need to do is to just prevent disease transmission like we're doing now. Make sure that we wash our hands, and if we've got a cold, stay home and try to stay away from others who have got.
So in this amazing way, maybe this pandemic that has really affected our whole lives, maybe there will start to be those little tiny silver linings for ecology and for the environment.
Yeah, I think so. And I think, that the whole kind of behaviour around stopping disease transmission will hopefully roll on to other diseases. So,
well, let's go on.
Yeah, but unfortunately what we're finding is in some of those severe coronavirus cases is that they are picking up secondary bacterial infections that are resistant. So in some scenarios, it's exacerbating the problem.
Let's get on to Scooper Poop, because the project, the concept, now I've been around little people for a while now in my different roles. They love talking about poo. I've also been around scientists for the last 3, 4, 5 years, working here at the Woodlands and Wetlands Trust, who love talking about poo. So when you came up with this idea to get families and kids involved with scientists scooping pooping, what an amazing idea. How'd you come up with the idea, Michelle?
I'm not sure if I should be saying this on national broadcast. No, I think it was at a conference actually, and I'm really not sure how it was coined, but I was just sitting there thinking about the challenges of surveying wildlife and getting enough samples that we need. And as a small research group, you can't do that, and you also can't sample broad areas. And it just kind of came about. I mentioned it to a colleague, and they came up with part of the app, which is called the Scatless, which is where anyone who participates, anyone can download the app, but you can actually go into the app and you can see the results from all the samples that people have collected and track your own. And we kind of thought the Scatless, Atlas, Scatless, if you're not quite picking up on that.
Yes, I loved that, yeah.
Yeah, and then we just thought about it, and then all of a sudden, Inspiring Australia had their first round of citizen science grants, and we were ready to fly. And it was
fantastic. All right, so talk us through it. How does Scooper Poop work?
Yeah, okay. So in non-coronavirus times, what we would do is we would go into a school or to a community group and we do a presentation and actually discuss the issues of antibiotic resistance. And then we go through the scientific process and demonstrate how the kits work, how people can get involved in science, how to use the characteristics of scat samples to make sure that you're collecting a brush-tailed possum sample.
And that's important, isn't it? It has to be a brush-tailed possum. It can't be other possums.
No, it can be a ringtail. That's okay. Yep. It's interesting because we started out that it was possums because of their widespread across different states and also from their occurrence in cities where we might expect to see a higher frequency of resistance. But we've also been working with some schools in rural areas and distance education and up in far north Queensland where they don't have possums. So we've also broadened our sampling to make sure that everyone can be included in the project. And it's, it works for the science as well.
Yeah, of course. So what do you need to have, what do you need to make it work or to be part of it? What do you need to have? What's in the kit?
The app, and then the kit, and the kit comes with, the kit, and the kit has some gloves, it's got a scoop of poop jar, it's got a barcode so you can label the sample and then you geolocate so we know where the sample's been collected. We don't know who collected it, just where it's been collected. And then the sample gets sent back to us and we do the tests in the lab.
And if people want the kits at this stage at the Woodlands and Wetlands Trust, we're working with you guys to get kits out to community groups. So
we are indeed. So in the ACT, definitely. And the project's going on for some time, so we can keep sending kits down.
Yeah, fantastic. Why is fresh poo important? So I'm thinking, if it's a daycare, really want to get involved, but the kids don't get there, you want to start collecting at 1 o'clock. Is it better to go first thing in the morning?
Oh, yeah. In cooler times, it is better to go first thing in the morning because it's probably it's actually a little bit easier to identify them. As if we're in 35, 40 degrees, they're going to dry out pretty quickly.
Yes.
We're actually testing the DNA. We're not isolating bacteria. But it also it's more about identification and So daycare centres could still collect them later on in the day.
Is there any risk that we can get sick from the possum poo at all when we're collecting it? Yeah,
so there are there's risks from any sample, any scat sample or fecal sample, our cats, our dogs. So the Scoop and Poop kit comes with gloves. And if you watch the videos on the website, we talk about making sure you wash your hands thoroughly. We show how to take the gloves off so you don't touch the outside. And if we're talking about young children, we always ask that the parents go along and they collect supervised.
And now at the moment, everybody knows how to wash their hands. So
exactly.
No problems there. When I was watching the video yesterday and seeing wash your hands for 20 seconds, I thought, yep, we're all across that.
You've got to get in.
Know what to do. Something that obviously scientists and our health professionals have known for years. Now we're all onto it. So once you get the samples, so we get our samples, we run around Canberra and Australia collecting them. What do you do with them when we send them back? What's happening in the lab?
Yeah, so they come into the lab and the first thing we do is a QC control and make sure that it is possum poop. Sometimes we've had grass or little bits of dirt. And then once
people thinking that's the poo.
Yeah.
Yes, fair enough. Yeah.
Yes. And then once we've done that, we then take very small amount and we extract the DNA. And basically the DNA, we then test that for the resistance genes. And the resistance genes are carried by the bacteria. I think this is a bit of a confusion that sometimes people make, is that they think the animal or us are resistant to the antibiotics. It's not, it's the bacteria that are resistant. So we're testing the DNA in the bacteria. Mm-hmm. So we do a test, see if it's present, and then we can do DNA sequencing and work out what antibiotics the bacteria in the wildlife species are resistant to.
And how long are you going to run the project for, the Citizen Science Project?
Yeah, the Citizen Science part, the current version of the Citizen Science Project is coming to an end in the next few months. So it's been running for 2.5 years now. So we've saved the grand finale for the ACT. So
wonderful.
Yep. And then the research will continue. and interestingly, the tools and techniques that we've developed and the kit is actually going to fold into a new grant that I've gotten with collaborator Claire MacArthur at the University of Sydney from the Australian, well, that Claire's together, we've got a grant yet. And that is all around understanding reverse zoonoses and pathogens in possums and extending some of the tools that we've got here, which is great as well.
I think it's such an amazing idea to get, as we say, citizen scientists, but getting kids and families and adults on board that might not have anything to do with science. And suddenly here we are all helping with a really important issue, tackling antibiotic resistance in the wild. But some of your work that you've done in the past, you've been looking at this for a long time. Can you tell us a bit about the captive breeding and that sort of silent threat that happens in captive breeding and wildlife conservation? Some of your work you've done in the past, it's pretty amazing what you do just by looking through the poop samples that you did.
Yeah, so the antibiotic resistance genes are an indication of wildlife picking up bacteria that aren't in their natural habitats, okay? So when we have animals in captivity or even in cities, they're exposed to the microorganisms that humans carry, but also that other species that are outside, so there might be birds in cities, or there might be a tiger next to them, or, not, maybe that's a bit extreme, but...
Well, I guess in the zoo though, you can have
in a zoo,
zoos.
Yeah. And so we've been studying, I guess, wildlife microbiomes and parasites of animals that are in captivity, and then comparing that to their wild counterparts to see if there's any risk of transmission throughout the conservation management program. So, and that's what we did with the brush-tailed rock wallaby. And we've been doing that with the Tasmanian devil as well. So yeah.
Because when you think of captive breeding, you think this is such a key factor in trying to conserve some of these species. What happened when you tested the brush-tailed wallabies that had been in a captive breeding program versus the ones that were in the wild? What did you find?
So we found the antibiotic resistance genes in the captive population, and there were none in their wild counterparts. And so had those animals been, well, it's kind of twofold. So I guess beyond that, just the presence of those resistance genes doesn't mean that there's necessarily a threat when those animals are released, or if those animals are released. But if those resistance genes are in a bacteria that causes disease, then they can potentially take that resistance gene, or they also act as an indicator that other organisms might be present. So there is a move in the, wildlife conservation and management now to actually think about what pathogens might be present and how to protect the health of their free-range counterparts when animals are released.
And it was really interesting when I was going through your paper and finding that there's a lot of translocation of wildlife, but not much disease screening. Is that still current, where there's not a lot of guidelines around it at the moment?
No. Yeah, I think certain programs are getting better at doing that, and I'm sure there's some recommendations and guidelines in different areas, but I'm not 100% sure the extent of those guidelines now.
Yeah, if it's changed.
Well,
it's just amazing what you can learn by looking at some poop. I think it's such a great, amazing work that you're doing, and we're so grateful for it. Michelle, thank you so much for joining us today on Bettong Bungalow to talk all things scooper poop and some of your research. And I know that...
I'll leave you with some...
fantastic. Are these some of the animals...
Wildlife there. I'll just change it over. Mix it up a bit.
Yeah. Tell us about, just before we finish, tell us about, are these some of the animals you've worked with in the past?
Yeah, so I was fortunate enough to go to Antarctica and again ask the same questions around the distribution of antimicrobial resistant bacteria. And so we were looking at the environment there and also a range of wildlife. And although we didn't find resistance genes in the wildlife, we found it in the water down there. And so that work is still important foundation work, and hopefully we'll get back to look at the wildlife in future.
Well, I think we'll have to have you on for another Bettong Bungalow, because now I want to learn about Antarctica and your work there. Thank you so much, Michelle. And I'm sure that people will be flocking to our office here in Ford to pick up their Scooper Poop kits and check it out.
That's fantastic. We've got hundreds that we can send down that way.
Brilliant. Excellent. Michelle Power has been my guest today on Bettong Bungalow. Michelle is Associate Professor at the Department of Biological Sciences, Macquarie University. We all know the title that she really loves is Leader of Scooper Poop Team.
Thank you.